Look closely at this plasma concentration time profile for apropocene HCl, where we track how the drug's mass per volume shifts within the bloodstream over a 20-hour period. Bioavailability is the critical measurement of both the total extent and the specific rate at which an active ingredient enters systemic circulation, represented here by the area under the curve. Observe how the multiple plot lines closely track one another. This overlap indicates bioequivalence, signifying that these different formulations achieve a nearly identical peak concentration, or Cmax, at the same time interval, Tmax. We determine that two products are interchangeable only when their absorption profiles demonstrate this statistical parity in both speed and total physiological exposure. Ozer and his team pioneered the term physiological availability in 1945 to quantify how effectively a drug transitions from its dosage form into the body's internal environment. We define bioavailability through two primary metrics: the relative amount of the dose that successfully navigates physiological barriers, and the velocity or rate at which this entry into the systemic circulation happens. Focus your attention on the precise interplay between mass and time. Not enough for the drug to simply arrive, as it must arrive at a speed and volume that maintains therapeutic relevance. Consider the implications if a high total amount of drug reaches the blood, but the rate of absorption is too sluggish to achieve the required clinical effect. Analyze these three fundamental pillars, starting with bioavailability, which quantifies the precise rate and extent of an active moiety's entry into the systemic circulation. The drug substance, or API, represents the specific chemical component that drives all pharmacodynamic activity within the biological target. Contrast this with the drug product, which is the finished delivery vehicle combining the API with inactive excipients to form a stable dosage form. How might the structural integrity of the drug product dictate the velocity at which the drug substance is released? This distinction ensures we account for both the molecular efficacy of the ingredient and the engineering of the delivery system. A brand drug product represents the initial synthesis and developmental stage, where a pharmaceutical entity holds a legal monopoly through patent protection. This patent serves as a temporary regulatory barrier, preventing other organizations from replicating the specific chemical formulation for a set duration. Once this protection interval lapses, the molecule enters the public domain, allowing for the creation of generic drug products by secondary manufacturers. Have you considered how the expiration of these intellectual property rights fundamentally alters the market availability of a validated chemical entity without changing its molecular structure? Every novel molecular entity must navigate this sequential regulatory architecture to prove its safety and efficacy profile before it can be prescribed to patients. Initial preclinical data regarding toxicity and pharmacodynamics are formalized in an investigational new drug application, which serves as the gatekeeper for transitioning from non-human models to human testing. The clinical phases then scale upward. Phase 1 assesses metabolic safety and dose ranging. Phase 2 targets therapeutic efficacy in a limited group, and Phase 3 validates these outcomes in massive, statistically significant patient cohorts. Following the submission and approval of the new drug application, Phase 4 surveillance begins as a longitudinal study to monitor long-term outcomes in the general population. How do you think the shift from controlled clinical environments to broad real-world use in Phase 4 might reveal rare adverse reactions not seen in the earlier trials? The Investigational New Drug Application, or IND, acts as the definitive scientific bridge that transitions a molecule from preclinical validation to human clinical trials. It necessitates a comprehensive synthesis of all pharmacological and toxicological data to demonstrate to the FDA that the substance is sufficiently stable and safe for initial human exposure. What necessitates such a stringent review of manufacturing controls and animal safety profiles before the very first dose is administered? This mandatory approval process ensures that the transition from a chemical compound to a biological intervention is governed by strict methodological integrity. Validation of an investigational new drug requires an exhaustive characterization of physical properties, like particle size and solubility, as these specific parameters dictate how effectively the substance will dissolve and interact with biological fluids. Rigorous analytical protocols must be established to quantify purity and concentration levels, because even minute fluctuations in chemical stability can drastically alter the safety profile before the first human dose. Furthermore, the submission must detail the formulation chemistry, explaining how the active moiety is integrated with specific excipients to ensure the stability of the final pill or liquid delivery system. Could a slight variation in moisture content or manufacturing technique undermine the entire pharmacological integrity of the product? Clinical trials progress through a rigorous hierarchical structure, beginning with Phase 1, where we evaluate pharmacokinetic parameters and dose-limiting toxicities in a small cohort of healthy volunteers. The focus then shifts to Phase 2, where we seek to establish the first evidence of therapeutic efficacy and optimal dosing within a controlled group of patients suffering from the target pathology. Phase 3 expands this inquiry into massive, statistically powered populations to compare the new against existing standard of care treatments, providing the definitive data necessary for a new drug application review. Once the regulatory body grants approval based on this comprehensive evidence, Phase 4 begins as longitudinal post-market surveillance to monitor for rare adverse drug reactions in the general population. In your view, why is the shift from the homogeneous environment of healthy volunteers in Phase 1 to the high biological variability of thousands of patients in Phase 3 the most critical step for validating drug safety? Once the cumulative data from Phase 3 demonstrates clinical success, the manufacturer submits the new drug application, the definitive administrative petition for marketing authorization. The FDA's interdisciplinary review team meticulously investigates the pharmacological evidence to verify that the safety and efficacy profiles justify the drug's entry into the public market. This scrutiny extends to the physical manufacturing infrastructure, where on-site inspections ensure the facility can maintain molecular consistency and purity at a commercial scale. Furthermore, the agency validates the precise scientific language on the drug label to guarantee that every dosage instruction and adverse reaction is accurately reported to the medical community. How do you think the rigorous verification of manufacturing facilities serves as the final barrier to ensuring patient safety beyond the chemical formulation itself? The abbreviated New Drug Application, or ANDA, streamlines the regulatory pathway for generics by removing the requirement for new clinical safety and efficacy data, focusing instead on proving bioequivalence to the original formulation. To achieve approval, a generic must demonstrate identicality to the reference-listed drug, or RLD, across 4 strict parameters: the physical dosage form, the exact milligram strength, the physiological route of administration, and the clinical intended use. This rigorous alignment ensures that the pharmacological response is predictable and interchangeable with the innovator product. All of these validated therapeutic equivalents are cataloged in the FDA's Orange Book, which serves as the definitive scientific registry for pharmacists and clinicians. Does the reliance on RLD parameters, rather than new clinical trials, imply any less rigor in the chemical validation of these generic counterparts? The abbreviated New Drug Application, or ANDA, is termed abbreviated specifically because it omits the requirement for redundant preclinical and clinical trials to reestablish safety and efficacy profiles. Instead, the scientific burden shifts to proving bioequivalence, where the generic product must mirror the performance of the innovator drug with mathematical precision. You are required to demonstrate that the active ingredient reaches the systemic circulation such that both the rate and the total extent of absorption are statistically identical. This means the velocity at which the molecule enters the blood and the total mass that eventually arrives must match the reference drug perfectly. How might a discrepancy in the manufacturing excipients disrupt this delicate balance of rate and extent, even if the active chemical remains the same? Relative bioavailability, often called apparent bioavailability, measures the systemic exposure of one drug product by directly benchmarking it against another. Mathematically, we express this as a ratio where the area under the curve of product A is divided by the area under the curve of reference product B, providing a precise metric of comparative absorption. If the administered mass varies between these two products, we must apply a correction factor to normalize the data for an accurate scientific assessment. In this scenario, you calculate the ratio by dividing the AUC of product A by its specific dose, then dividing that result by the dose-normalized AUC of reference B. Why is this normalization step absolutely vital when comparing products that aren't identical in milligram strength? Beyond plasma analysis, we can precisely quantify relative bioavailability by measuring the total mass of intact drug recovered from the renal system. The mathematical core of this approach is the ratio of cumulative urinary excretion values, where we take the total amount of drug excreted from formulation A, denoted as the bracketed du subscript A at time infinity, and divided by the total amount excreted from the reference product B, or bracketed du subscript B at time infinity. It is essential to ensure that the collection period spans a sufficient duration to reach this theoretical infinity, signifying the point where the kidneys have completed the elimination of the active moiety. This technique essentially uses the bladder as an organic storage tank to integrate the total systemic exposure over time. If a patient's renal clearance fluctuates significantly between the two administrations, how would that inconsistency compromise the statistical validity of your final ratio? Absolute bioavailability, denoted as F, determines the precise efficiency of a drug's absorption by benchmarking it against the total systemic entry of an intravenous injection. Examine the equation where we divide the dose-normalized oral area under the curve, AUCPO, over its dose, by the dose-normalized intravenous AUC, AUCIV, over its dose. This ratio effectively cancels out dosage differences to isolate the exact proportion of the active moiety that survives the transit into the bloodstream. Why does a final value significantly lower than 1.0 indicate the presence of a high first-pass effect or significant physiological barriers? You will define this metric either as a decimal fraction or multiply it by 100 to express the absolute percentage of the administered dose that reaches systemic circulation. We are now determining absolute bioavailability by analyzing the total mass of drug excreted through the kidneys, rather than measuring plasma levels. To calculate the bioavailable fraction F, you must divide the dose-normalized urinary recovery from an oral administration, represented as DUPO at infinity over the oral dose, by the dose-normalized recovery from an intravenous injection. This equation establishes the IV route as the gold standard, where F equals exactly 1, while all extravascular routes will mathematically result in an F value less than or equal to 1. How might a significant decrease in a patient's glomerular filtration rate between the two study periods disrupt the precision of this ratio? Let's synthesize our understanding by solving for the bioavailability of this antibiotic across 4 distinct delivery systems. To determine the absolute bioavailability of the tablet, you must divide its dose-normalized urinary recovery, 340 over 4, by the IV reference's dose-normalized recovery, 20 over 0.2, resulting in a fractional value of 0.85. When calculating the relative bioavailability of the capsule versus the oral solution, we simply divide their urinary excretion values at infinity, 360 by 380, to find a comparative efficiency of 94.7%. Why does the consistency in dosing across these oral products allow us to omit the dose normalization step in this specific calculation? To quantify how a drug enters the system, we rely on 4 fundamental methodologies: plasma analysis, urinary excretion studies, acute pharmacologic response, and in vitro dissolution. In plasma studies, we pinpoint Tmax to determine the time required to reach peak levels, Cmax for the highest concentration achieved in the blood, and the area under the curve to integrate the total systemic exposure. Alternatively, urine data allows us to calculate du over dt, which represents the instantaneous rate of drug excretion, alongside du infinity, the total cumulative mass of the drug eventually eliminated by the kidneys. These direct measurements in biological fluids provide our most objective data because they track the actual movement of the active moiety, rather than relying on subjective clinical observations or external dissolution tests. What factors do you think make measuring the chemical concentration itself more reliable than simply observing the final pharmacologic effect on the patient? Tmax identifies the specific moment in time when the rate of drug absorption exactly equals the rate of drug elimination, resulting in the peak plasma concentration seen at Cpmax. This value acts as a critical diagnostic for absorption kinetics. The more rapidly an active moiety crosses biological membranes into the systemic circulation, the lower the Tmax value becomes. Measured in temporal units like hours or minutes, it allows us to quantify the speed of the delivery process independently of the total dose administered. If we were to observe 2 different formulations of the same drug, how would a significantly shortened Tmax in one indicate a more efficient disintegration or dissolution profile? Observe the pinnacle of this curve, labeled Cmax, which defines the absolute peak plasma concentration achieved following a single oral administration. You must express this value using precise mass over volume units, such as micrograms per milliliter, to accurately quantify the highest level of systemic exposure. Critically, Cmax must fall within the therapeutic range. If it drops too low, the drug is ineffective, and if it climbs too high, it becomes toxic. Based on this relationship, why is a formulation that causes Cmax to spike rapidly into the toxic zone considered a failure in pharmaceutical engineering? The area under the curve, or AUC, serves as a geometric representation of the total systemic exposure, integrating the plasma concentration from the moment of administration until the drug is completely eliminated. By calculating the mathematical integral of this plot, we determine the extent of bioavailability, which specifically quantifies the cumulative mass of the active ingredient that successfully enters the bloodstream. You must express this metric in units of concentration multiplied by time, such as microgram hours per milliliter, which represents the mathematical product of the y-axis mass and the x-axis duration. If you compare two different formulations and notice one covers a much larger shaded region despite having an identical Cmax, what does that reveal to you about the total physiological exposure provided by that specific product? Observe the progression on the left as the dose scales from 250 milligrams to 1,000 milligrams. The corresponding plasma concentration time profiles expand symmetrically, reflecting that the total systemic exposure is a direct mathematical consequence of the mass administered. This proportionality is solidified in the right-hand graph, where the plot of area under the curve against dose yields a consistent linear slope, governed by the formula where AUC equals the bioavailable dose divided by clearance. Because this line remains straight and passes through the origin, we can conclude that the physiological processes of absorption and elimination are operating well below their saturation limits. What specific change in the slope of this line would suggest that the body's metabolic enzymes have reached their maximum capacity and can no longer process the drug at a constant rate? We shift our focus to urine data as a robust alternative for estimating bioavailability, which requires that a statistically significant portion of the active drug is excreted unchanged through the renal system. To interpret this data, you must master two key parameters. First, the cumulative amount excreted at time infinity, or due infinity, which directly mirrors the total extent of drug absorption into the body. Second, we analyze d due dt, a mathematical derivative representing the instantaneous rate of urinary excretion, which serves as our proxy for the drug's absorption velocity. Why do you think using a drug that is almost entirely metabolized by the liver before reaching the kidneys would render these specific urinary calculations invalid? By aligning the plasma level graph with the cumulative urine profile, you can see that at point C, the plasma concentration approaches zero, signifying that the body has finished processing the dose. This point C is where the lower graph plateaus at D infinity, which is the mathematical total of all drug mass recovered from the kidneys, representing the total extent of absorption. To determine the speed of the drug's entry, we calculate d du by dt, the instantaneous derivative or change in urine amount over time, because this rate of urinary output directly tracks the rate of absorption into the systemic circulation. If you observe the steepness of the curve between points A and B, why does a more vertical slope in the cumulative urine data necessarily translate to a higher peak concentration on the upper plasma graph? Look at these three distinct plasma level profiles, and notice how the geometry of the curves defines their pharmacological behavior. When you see that the area under the curve for A equals that of B, you are verifying that these two formulations achieve the exact same total extent of drug absorption into the bloodstream. However, because the time to reach peak concentration, or Tmax, for A, is less than that of B, we can conclude that formulation A possesses a much faster absorption rate. Shifting to formulation C, the equation AUCC equals 0.5 times AUCA reveals that this version delivers exactly half the total systemic exposure of A. Interestingly, since the Tmax for A and C are perfectly aligned on the horizontal axis, how would you justify the conclusion that these two products have identical absorption velocities despite the massive difference in total drug delivered? Analyze these plasma concentration thresholds where the horizontal line MEC represents the baseline concentration required for clinical efficacy, while MTC marks the ceiling where toxic side effects begin. Formulations A and B are both therapeutically successful because their peak concentrations, or Cmax values, fall precisely within this therapeutic window, though formulation B provides a more prolonged duration of action due to its slower absorption rate. In contrast, examine curve C. It is a clinical failure because its Cmax is mathematically less than the MEC, meaning the active moiety never reaches a high enough density in the blood to activate its biological targets. If we were to increase the dose of formulation C, what would happen to the relationship between its new Cmax and that MEC threshold? And could we eventually push it into the therapeutic range without redesigning the delivery system itself? Focus on the top left graph. Formulation A exhibits a much steeper initial slope and higher Cmax compared to B, indicating a significantly faster rate of absorption and a greater total extent of drug reaching the systemic circulation. Observe the top right scenario, where the peak concentrations are numerically identical, but the horizontal displacement of the Tmax markers shows that A reaches that peak much sooner than B, proving a difference in kinetic rate despite similar peak heights. Looking at the bottom plot, both formulations achieve their maximum concentration at the exact same timestamp, yet the vertical magnitude of A far exceeds that of B. This alignment demonstrates that while the absorption velocity is perfectly synchronized, the total bioavailable mass of A is significantly higher than B. Which specific geometric change among these three would most likely indicate a failure in the drug's sustained release coding versus a simple reduction in the total milligram dose? We are examining the acute pharmacological effect, or the acute pharmacodynamic effect, which allows us to measure bioavailability by monitoring the body's direct physiological response to a dose. This specific strategy is employed only when a reliable chemical assay is unavailable, meaning you cannot accurately or consistently detect the drug's concentration within plasma or urine samples. By quantifying precise changes in parameters like pupil diameter, heart rate, or blood pressure, we can mathematically correlate these biological shifts to the drug's absorption and distribution. If the analytical equipment fails to find the molecule itself, how can we use the sudden onset of a biological effect to validate the speed at which the active moiety reached its target receptor? This method essentially transforms the patient's own cardiovascular or neurological systems into a living, real-time detector for the drug's systemic presence. Shift your focus from biological fluids to the laboratory environment, where we use in vitro dissolution studies as a predictive tool for how a drug might behave inside a living organism. Scientific data generally demonstrates a direct linear relationship between these environments. A formulation that exhibits a rapid dissolution rate in a test beaker will typically manifest a high rate of bioavailability in vivo. Our ultimate objective in pharmaceutical development is to establish a rigorous in vitro in vivo correlation, or IVIVC, where the laboratory dissolution profile mathematically predicts the actual plasma concentration time curve. Achieving a validated IVIVC means that we can rely on benchtop experiments to guarantee that any manufacturing modification will not compromise the therapeutic performance of the drug in the patient.